Many people have contributed to the work in here. I've just sort of cherry-picked many slides in an attempt to provide, I guess, an overview of next-gen sequencing as I hope relevant for people at the Broad. So this is my self-introduction. I'm currently the manager of medical and population genetics analysis in the medical and population genetics program. As part of that, I run a group called Genome Sequencing and Analysis. We do a lot of projects here at the Broad. This is the 1,000 Genomes Project. We do a lot of the medical genetics projects. We develop a toolkit called the JATK. Before I joined the Broad, I did a variety of things. I mean, one thing, I was actually a business consultant for a while here at a local consulting firm. I was a Runyon Fellow at Harvard, actually doing experimental evolution. So I was an experimentalist before this. And before that, I was a biochemist. And then even before that, I was a mathematician. So I guess I've worn many hats. And ultimately, though, they led me to here, which is sort of a good combination of experimental work and computational work. So I think the key goal of this is not really to provide any particularly deep understanding of any one topic, but an attempt to give like an overview of a very complex process that goes from preparing a library to actually having a publication where you discover some new causal variant. And this part of I'm sure that there'll be other talks in detail about, given some variants, how do you figure out what are causal alleles. But I really wanted to try to take us from the first part of that. So you have an experiment or some sort of question you want to answer. You're going to use next-gen sequencing to do that for detecting genetic variation. Sort of what are the parameters to think about when you're doing that, and what does the overall sort of process look like? So really, this is broken down in a couple of parts. I mean, first thing we got to talk about is machines. Most of this is going to be terminology. I hope it'll be useful. These are slides that I wish I had seen when I joined the Broad, so someone would have explained to me what some of the terms meant. Also, some of the processing steps, basic things we already understand, and open issues. And finally, I think a critical thing of understanding how to assess what you're getting from these machines. So I think this is actually a very useful slide. This is from a paper that we are about to resubmit. It should be, I guess, accepted soon. This is sort of a high-level overview of what are the steps involved in analyzing next-gen sequencing data. And it's really 3 phases. It's useful to even understand that there are 3 conceptually different phases as opposed to some one giant monolithic process. The first phase is really raw data processing. I mean, the machine runs, it generates these reads. They're raw. There's very little information associated with them besides a bunch of bases and a bunch of estimated quality scores. You have to take this enormous amount of data and map it to your genome for resequencing projects. You need to do some sort of local realignment to ensure that there's consistency among the data at each site. You need to figure out which reads are actually molecular duplicates of each other, which is a critical thing if you have a low complexity library. You need to actually figure out what the error mode is per base. And at the end of that, you have something that, you have a big pile of reads that are aligned to a genome that have a well-calibrated error process and are mutually self-consistent. So you have what we think of as an analysis-ready read set or analysis-ready BAM. And at that stage, you're doing that per sample. You then have many samples, potentially. You're sequencing your family that has a Mendelian disorder. You have a couple of samples. Maybe you're part of the ESP project where we're doing 10,000 exomes. Ultimately, though, you have some number of samples that you have reads for, and you're going to produce a raw list of variants. So this is going to be SNPs or Indels, structural variation. And these would be all sites that could potentially be variable. There's some evidence in the data such that you could have a SNP there, or you could have a machine artifact. That's why I call them raw. There's no attempt at this stage to strongly differentiate between sites that are true polymorphisms in a population and sites that are just machine artifacts. And finally, at this sort of third phase, this integrative analysis, you're really taking raw SNPs, raw indels, the raw structural variants, and bringing together lots of information. The pedigree that you happen to know about from your study design, maybe the population structure. You have Africans and African Americans and Europeans in your group. You have known variation. 1,000 Genomes has produced a list of 25 million sites that are known to be polymorphic in humans. Maybe you have genotype data from GWAS studies that you've done. And you're taking all of that data and attempting to integrate it in to get a single estimate of the probability for each of these raw variants, what the chances are that it's a real variant and not a machine artifact. And you're typically cycling that with some sort of imputation or genotype refinement, something to give you the best possible picture of the probability that each site in the genome is polymorphic. And then if it is polymorphic, what are the genotypes of all the samples in your study at that site? That's sort of the overall picture at which point you could go and actually start doing some analysis. So let's just start with some basic terminology here. Hold on, good, okay, good. I just need to see a clock, there we go. So I'll go very quickly through this introduction. This stuff will be available on the web for everyone who wants to look in detail at this. Basically, this is what people call next-gen sequencing. There's 454, there's Illumina, there's Solid. There are 3 machines of approximately equivalent quality. 454 is a little more expensive these days, but these are the 3 major players in next-gen sequencing. This is in contrast to early ABI days. This is sort of the original Star Trek cast. Then there's the 3730s, the workhorses of the Human Genome Project. This is really still old school Star Trek. I grabbed these slides from somebody. I don't know. If you're a Star Trek person, this is a good analogy. I'm sort of struggling here with, I don't even know what this next image is, but this is a helicose machine. This is sort of a single molecule sequencer. It's sort of, I don't know, generation 2.5. It's very similar to Illumina and Solid and 454, but is actually working at single molecule level. And finally, there's these sort of next generation machines. It's unclear. I hope they're not called next generation from now on. I think they'll probably be 3rd generation machines. There's the things like Pacific Biosciences machine, Nanopore, Ion Torrent. These are machines that have, I think, potentially radically better capabilities than those next generation machines, but are not yet at the scale of either 454, Illumina, or Solid. So to give you an idea of what we're struggling with here in terms of data production, this is, I think, a very nice figure that Stacey Gabriel gave me here that is the number of gigabases produced by the Broad as a function of the number of the year. So this is looking good, 50 gigabases, 60 gigabases by 2006. You then move a couple of years out as the next generation machines start to arrive, and you immediately go up by orders of magnitude in the production of sequencing data here at the Broad. And then now you're here where we're producing an enormous amount of data relative to what we even did a couple of years ago. And the next generation machines have a sort of a promise to push this curve continually up at the same sort of hyper exponential rate. So today's workhorse at the Broad is this Illumina HiSeq 2000. This just gives you an idea of some technical details of the machine. Basically, it's 100 bases or 76 base reads, all paired end, very low error rates, very high yield per run. So the critical number here is really yield per run in gigabases. So a GA2X, which is a pretty advanced Illumina machine, is producing 50 gigabase runs. The HiSeq is already at 250, and there's talk about it going to 500 within 6 months. To give you an idea, 500 gigabases, there's 3 gigabases in a human genome. So this machine is producing around 80x coverage of a human genome per run. So it's a lot of data. So just starting to step back now that we have some idea of what the landscape looks like, I just wanted to define some terms, because you will hear people throw these around, and they're critical for understanding the next-gen analyses, the data processing tools, and really what experiments are being done. So a library is really a collection of DNA fragments that has been prepared some way. So this is an example of a paired end library prep. It's producing a library of DNA molecules. So, this is really the sequencing unit. I mean, you are putting a library of molecules onto a machine and sequencing it. So the example is, if I have 1 million molecules in my library, I don't know, maybe I diluted it down, I can only sequence 1 million unique fragments from that library, no matter how many reads I happen to get. They start to sequence the same molecule. So you can have libraries that have different complexities. You can have different preparations of libraries given the same sample. So maybe one library has 400 base fragments. Some have 2,000 base fragments. So this is what they're really talking about different libraries can be, or they're just different DNA preps from the same sample. Then the other key units are flow cells and lanes. So a flow cell is really one of these units of the machine. I guess it's really it's a chip here that's being run on the same machine. And it has multiple lanes, each of which is sort of a unique sequencing unit. So people are typically talking about libraries and then lanes sequenced. So you'll talk about, I have my exome, I did one library prep, it was captured, and then I sequenced that library on 3 lanes of the HiSeq to get my final data set. That's sort of a standard description of a data set. So the other things to appreciate, wow, this is intolerably difficult to see. I don't understand why that would be. I will gesture then. So there are actual objects here in the screen. There are these, this is just, all right. You'll have to look at this later online. The slides are all in PDF form. Basically, the point of this slide was to differentiate 2 things that are critical. So there are reads. Reads are just the raw units produced by the machine. It's just sort of a sequence of bases and quality scores emitted by one of the machines. So we talk about a 100 base read. It's 100 bases that came off the machine independently of all the other reads. A fragment is really the original molecule that is being sequenced and emitted as that read. So you can have multiple reads sequencing the same fragment. In fact, when they talk about paired end reads, what they're really talking about is 2 reads that are reading from different directions of the same fragment. So in fact, the only thing that you can even see in this image are the first and second reads from the same fragment. So there are some critical things to understand here that I think are useful. You can sequence the same underlying molecule twice. That's what you see in the reads that are going, these purple reads that are overlapping. The parts that overlap are actually observations of the same underlying DNA molecule twice. So not all reads are independent. Really, the things that are independent are the fragments. Good. So,
can you talk about the
sure.
Cluster and how many little fragments are in clusters?
So a cluster is an individual read being, it's really, so the way these machines are working, you have a slide, and you've sort of bound your DNA molecule, your fragment to the cluster, to the slide. And then you start to sequence on that. And the cluster is really an amplification of a single fragment that is being observed by the machine. So they can, I can't remember exactly the exact details. It varies by machine, exactly how they get the paired end. So you're bending over typically the fragment to another part of the slide and sequencing that, and that would be a different cluster that you're observing.
So is there like 1,000 reads in a cluster? So a fragment?
No, a cluster, well, it depends on whether you're single molecule or not. So a machine like an Illumina, there are many PCR amplicons immediately adjacent to the original fragment that compose the cluster, because you need to amplify the signal that you're viewing on the slide. So you think about it, you have a camera that's looking at a fluorescent image. And in order to have enough signal to see the fluorophores that are showing you which base is there, you have to either have an incredibly sophisticated camera that can observe single molecules, which is what you would get with a helicose machine, or you would have to amplify the signal somehow by amplifying the molecule locally, and then you would get a homogeneous fluorescence image in the neighborhood of the cluster. So it varies a little bit. If you have single molecule, basically summary is if you have single molecule sequencing, there's no amplification. If you have many of the technologies are doing a local amplification on the slide so that you can amplify the signal that's observed for which bases are there. I'm happy to talk more in detail after this thing. So I want to talk a little bit about how are we thinking about experimental design in this, given what the current capabilities of the machines look like. And I threw this together fairly quickly because one way of thinking about it is, it's very unusual that you can do everything you want to do. Ideally, you would do many, many samples, and they would all be very deep. But typically, what you have is a trade-off. I have a fixed pot of money. I have some number of samples that I would like to sequence, and I have to make a trade-off between how many samples am I sequencing, how deep of coverage do I get per sample, and how much of the genome do I sequence? And so this is really the three, I think, characteristic sequencing designs you see being run in MPG now. The classic one is the deep single genome. So this is, I have a cancer normal and a tumor sample, and I sequence them both to sort of 30x, and I can do some comparison. I have very, very good ascertainment of the genome at every position because I have a lot of data everywhere. This is also very expensive. I'm doing nearly 50 or 60x coverage everywhere. Another way of doing whole genome and getting more samples for the same cost is really what the 1,000 Genomes has done in the Type 2 Go, which is to do less coverage per sample and do more samples. And this is really strong if you want to discover common variation. But of course, it comes at the cost that you really don't know exactly what's going on in any individual sample because you have less data to work with per sample. There are projects like the exomes, which are very, very popular designs here. I would say most of the samples that are being sequenced at the Broad now are exomes. Those exomes are really, there are many very deep, actually probably more deep than the single deep genomes. There are a lot of samples, and what you've done is you've reduced the target area. So you're only sequencing 1% of the genome, and that's why you can have a fixed pot of money that seems to give you many more samples and greater depth. It's because you're getting less data, because you're targeting less of the genome. And then there are these really candidate gene follow-up things like the Framingham project and the Pfizer project. These are a select set of genes, 100 or 200 or 300. You have a custom chip capturing specific genes. And because of that, you have a 1 megabase target or a 2 megabase target. And then you can do many more samples at higher depth for the same amount of money. So I just wanted to review quickly hybrid selection, what is going on with hybrid selection. This is what we're using for capturing selected regions of the genome. What you have are baits. These are oligonucleotides. They have a biotinylated tag, so you can actually grab a hold of them. You have a PON, this sort of genome library, as I was saying earlier. You then mix them together. You get the probes binding, well, the baits binding to your target sequences. You have the biotinylation, well, I can't remember what it is, biotinylation site to grab a hold of. You catch them, and then you sequence the diluted results of the capture with the baits of your whole genome. And this allows you to basically select particular areas of the genome for enrichment. And this is a pretty mature technology here at the Broad. Certainly, standard whole exome capture chips are extremely robust. Custom ones are more complicated. You have to be a little careful with how you do the design, but it's routine now to do this. So I just wanted to emphasize a couple more useful terms here, because you hear them being batted around, and it's important decisions. So there's a difference between genotyping and pooling strategies. Oh, go ahead.
To what extent can those beta strategies handle variation in genome inaccurately pulling down?
It's surprisingly robust. That is sort of my take on it. As far as I can tell, there's very little differential capture based on, for instance, having a heterozygous SNP. I mean, early captures, or if you do a design that has very short baits, this will hurt you. But the baits are 120 bases long, so they're pretty robust to single base variation. As you start to get larger and larger variation, it becomes more sensitive. But it's still pretty robust. If you ask what fraction of, for instance, indels, which you might expect would be really difficult to capture, affect the capture efficiency, it's clear that deletions have much less effect than insertions. It's only once you get to larger insertions are we really seeing an obvious effect of the lack of capture. And you can tell this because you get an asymmetry in the insertion deletion spectra. So you say, I have 100 one-base deletions and 100 one-base insertions, and I have 10 two-base deletions, but I only have two-base insertions. And this you get this skew in that distribution, which is not biologically reasonable. And so it's clear that that's affecting you, but it's pretty restricted. I mean, it's not as bad as one might guess. But if you're really interested in 20-base insertions, capturing is going to be difficult. So I think we'll just go through quickly the difference between genotyping strategies and pooling. So what does it mean to say, I've designed my project so that I'm going to be able to do genotyping? What this means is that you're preserving the relationship between reads and samples. So the example, this is exactly the same set of variants here. Some are heterozygous, some are homozygous. There are two samples on the left, and because I know the relationship between the red reads being from sample 1 and the blue reads being from sample 2, I can tell that somebody is a heterozygous, somebody's a homozygote. If I'm pooling those samples together and I don't know that relationship, I now do not know which reads come from which people, the best I can really say is that things are common or things are rare. But I don't know that somebody's a heterozygote or a homozygote for any individual variant, because I don't know. I can make an estimate, but it's not transparent from the data. The key is genotyping algorithms are much more powerful at analysis. If you have a design that preserves this relationship, you can apply multi-sample genotyping algorithms that work very, very well. If you have pooling strategy, it is harder to control error, and it's much harder to figure out what's going on. I'm happy to talk to people after who are interested in this. The problem is pooling is very attractive because it's cheaper, but it does make the analysis much harder. I'm going to skip over this in the interest of time. I'm also going to talk a little bit about single versus multi-sample analysis. This is an example here again. It's assuming that I have gone down the route of having a genotyping assay. So I have a relationship between reads and samples. I now can ask this sort of question of, like, given a fixed sequencing budget, what do I get out of the trade-off between spending that sequencing budget on getting more data per sample versus more samples but with less data per sample? And you get, again, very similar trade-off between the genotyping and the pooling. What's going on here is that you have much better ascertainment. I could tell very clearly which sites are heterozygous, which are homozygous in the single sample deep. It's harder to figure that out in the shallower multi-sample case. But the critical trade-off here is that I only find 3 variants in the deep sequencing, but I find 5 variants in the multi-sample case. So I'm trading off breadth of discovery for genotyping accuracy per sample. And depending on your goals, you may or may not want to be further on the left or the right. 1,000 genomes is very far on the right. We do 4x sequencing of 1,000 people. Cancer genomes are typically very far on the left. I want 40x coverage of a single individual because I want to absolutely find all the heterozygotes, and I want to know that it's heterozygous, not homozygous at the site. So given that, I want to go through this sort of the 3 classic sequencing study designs that you'll see right now. First is I think the easiest to understand. This is what's really mostly in the literature from 2008, 2009, these high-pass sequencing designs. This is a whole genome target, very deep coverage, 30x or greater. You basically have excellent sensitivity and excellent genotyping. And this gives you an idea of what it would require for each sample. So the data requirements per sample, you're targeting 3 gigabases of human genome. You want 30x coverage, you need 100 gigabases of sequence. This is 8 lanes on a high-seq right now. And then you say, how many variants would you find? You find around 3 to 5 million, depending on the ethnicity of your sample. You're going to find 99% of all the variation in the genome with 99% sensitivity to all singletons and all common variation. So this is a very easy design, and it's very reassuring. You get a lot of data per sample. Or sorry, you get excellent ascertainment for single samples. The next one is very similar, which is just the low-pass version of that. So instead of doing 30x, I do 4x. And so what ends up happening here? The heterozygotes, because of sampling, can be mistaken as homozygous. So it becomes harder and harder to genotype because of sampling problems. Sometimes just sampling will mean I miss a site. I stand a pretty good chance for a heterozygote at 4x coverage to just never see the alternate allele. I mean, the chance of not seeing any reads from a heterozygote at 4x coverage is like 25 or 30%, something like that. So you stand a very good chance of missing it entirely. So if you're looking for rare singletons or really rare variants, this is a very bad design for you. There's also, however...
I have a question about that.
Mm-hmm.
This 1,000 genome project is being used by a lot of people to establish normals.
Yeah.
And what you just said makes me a little worried about that.
Sure.
So how do you reconcile those It's the normals that become, if you're in the rare variant business,
Yeah.
real-ish.
Yes.
And so the 1,000 Genome Project is looked upon by most of us as at least a partial solution to that.
Right.
So how do you reconcile it?
What we've decided to do is to make a trade-off. So I could sequence many individuals to very deep coverage. I would have fantastic ascertainment of everything in those individuals. But I will miss everything that's actually exists out in other samples that I didn't sequence. So if you're asking how efficient, what is the most efficient design to capture the variation in the population, you would prefer to do shallow sequencing of the population because it allows you to discover things that are common and shared among the samples. You lose accuracy of really rare things, but anything that's shared, it's very easy to find. And this is a good example of that. So these guys, this only has 2x coverage per read, but the site that has occurs in 3 people is clear already. No matter how many other people I happen to sequence, as long as I see it 3 or 4 or 5 times, it's very easy to call. So you're trading off ascertainment in the allele frequency spectra for certainty in each individual. And you get a lot more variance just by doing shallow or multi-sample.
Another way of stating that is it favors people looking for common variants and puts that at disadvantage as rare variants.
Exactly. So this is exactly right. And in fact, you can see this right here. I mean, these are some numbers I estimated. One way of saying, given the low-pass design, I can sequence 5 individuals for every deep whole genome I do here. So you can do 5 people for every one. So it's quite a trade-off in terms of cost. But you can see what's going on here. I'm only going to find 90% of the variation in any individual genome in isolation, and my probability to discover singletons is below 50%.
Is there a plan to come back at 1,000 genomes as the capacity you showed the first slide rises and have your cake and eat it too?
We could, certainly. I mean, and also 4x is a particularly bad point in that trade-off. I mean, 6x is already doing very much better, and 8x is pretty close to as good as you get with deep coverage. I mean, it's not perfect, but if you're asking what is the marginal additional value, it goes up, it's very steep, and it saturates pretty quickly.
So it wouldn't be a big deal for them to go from 4 to 8x.
Most of our samples are above 4x. It's to the point where we measure our coverage, we have to cap the sequence in the averaging of the average coverage per population at 5, because so many samples are at 10. And so you don't want to skew your average coverage up. So this is enough of a problem already that people are just throwing the samples on sequencers. And we're getting 6, 8, 10x coverage in many of the new samples.
In the annotation when you report that, do you indicate the x coverage so that...
It's available. It'll show up also in much better ascertainment in that sample. And also because of the way the multi-sample calling works, it actually projects better, improved sensitivity and specificity into the other samples even at lower coverage. So I haven't talked in detail here. I'm happy to talk in more after the thing about, it's not obvious that the best strategy is all single low pass. There may be value in spiking in single, select individuals at high pass and then doing other samples at lower pass. Because once you know a site is variant, it doesn't take very much to be able to ascertain that it's variant in the other samples. So a question up there.
Just to follow up very quickly. So if we're screening variants against 1,000 genomes and the variant shows up as a heterozygous hit in one person, should we trust it or not?
It'll probably be pretty accurate. In fact, the ability to call a singleton at 4x coverage means you probably were higher you probably had higher coverage at that site. And then given that we've also applied imputation tools to do this, to preserve the singletons means that they're probably pretty good haplotypes in the area. So it's really more not an accuracy issue as an ascertainment problem. You're losing many of them, but the ones that come through are pretty accurate, I think.
Could you just expand on the haplotype block issue you just
Sure.
alluded to? Because that seems like another way to get around the problem by looking at that.
No, so we do. I mean, you're doing a very good job at, why don't I say this? I will come to a slide that describes the efficiency of this process. OK, let me just keep going here. This is done. Wait a second, I'm going backward. OK. OK, good. All right, so exome capture is just basically the trade-off between, this is high-pass sequencing, but in select regions. And the cost here is the obvious one. There's little off-target coverage. So you will miss things that aren't in your targeted regions. Sort of obvious. So let's talk about data processing quickly. I want to keep moving along. I'm happy to keep taking questions, but we have to finish in 30 minutes or so. So data processing, what does this look like? I showed this slide. I'm going to run very quickly through all three parts of this figure, just to give you an idea of what's involved. Commonly used tools, there are many effective tools for next-gen sequencing analysis. I would highly recommend that if you have new data and you're new to the field, to try out some of these tools before writing your own, say, snip caller or mapping algorithms. These things are pretty effective already. We develop here a tool called the GATK. A couple of the developers are up there if you want to ask them in detailed questions afterward. Basically, it's a toolkit and a set of tools for developing algorithms for next-gen sequencing. It has many very popular tools in it now. There's also a framework for doing, for actually building pipelines to execute any variety of tools in series. This is what we actually do for many of the projects here now that we're running. And the critical thing is it uses, we've been part of the development of these two key file formats, the SAM-BAM spec, which is to represent next-gen reads, and then the VCF format, which is becoming a standard for the calls. So just a plug here on BAM format. I couldn't recommend more strongly that if you have next-gen sequencing data, you get it into BAM format. The Broad already is using this extensively. This is more for reference for people. One thing that I would like to emphasize, we sort of hold a line of requiring pretty extensive header information in the BAM files. It's optional, but really essential. Critical things are, when I was talking about lanes, these are described as read groups in the SAM-BAM spec. It provides metadata that tell you from an individual BAM file what type of machine was used to produce that data, information about the lane that was run, and critically, the relationship between the reads and the lanes, because you may have multiple samples present in a single BAM file, and being able to disentangle reads so that you know which read came from which sample and which read came from which lane on a sequencer allows you to do analyses of multi-sample BAM files, which is really critical because single sample BAM files are becoming rarer and rarer, and you don't want to get in a situation where you have 10 terabytes of data and you don't know what the relationship between the reads and the data that the machine that was used to produce and which sample is in which BAM file. Just naming your BAM file sample1.bam is a bad idea. You might as well put it in the BAM file so it can't be lost. So what is going on here in terms of data processing? I mean, these are the key steps. So suppose somebody gives you a big pile of next-gen sequencing reads right off of a machine. First thing you do is convert it into SAM-BAM format, and then you map it to the genome. And there are many mapping algorithms. I would recommend looking at Hang Lee and Niels Homer's review here if you want more details. The critical thing that it does, and it is very simple, it takes the big pile of reads and figures out what the origin of the reads are from the reference genome, and critically tells you the probability that the reads actually go to that site. Because there's many sites in the genome that are ambiguous. They're identical in very short stretches. In the limit, if you only had a one base pair read, there would be a lot of A's or T's in the genome it could have come from. As you get longer reads, you have more specificity in that mapping, but there's still many areas that are very, very similar to each other. So it's important to differentiate between just placing the reads, which is easy. I can put them wherever I want, and actually telling me the probability that it came from there, which is really key. Wow. All right. I will skip over this. Suffice it to say that it's very important to deal with indels. There are a variety of things that it's very easy to mistake because these reads are very short. Indels for SNPs. There are many tools to deal with this. Also, the quality scores of the bases themselves are still inaccurate. What goes on here is that the machine is trying to give you an estimate of the confidence when it tells you that the base that it just read off the machine is an A. It tries to give you a probability that that's real, that it really is an A. And that probability is not necessarily particularly well calibrated. It's not terrible, but it's not 100% accurate. we have tools that not only will fix the empirical relationship, but also attempt to correct out covariates, things like the cycle of the machine, the cycle that the base was read in on the machine is a major determinant of the accuracy of the estimate. And so this base quality score recalibration is something that we've been developing for a long time, and it is a fairly popular tool now. And it's automatically run at the Broad. So your data would look like the blue lines, not the red lines, if you're using Broad-generated data. So how does SNP calling work? So now that suppose I've gone and I've done all this analysis, I've cleaned up my BAM files, I've recalibrated all the quality scores. You're basically doing two things. You're estimating the genotype likelihoods for a diploid individual at every site in the genome. So this is, I'm establishing what is the likelihood of the data, of the reads, under the hypothesis that the underlying genotype is A, C, blah, 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 blah. So I have 10 likelihoods. This is, in order to do some quality control, you want to restrict yourself to only good reads and a variety of other things. OK. Now, the next step of that is you take the individual genotype likelihoods per each of your samples, and then you're doing a simultaneous estimate of the probability of having exactly I segregating copies of an alternate allele among all your samples, and then establishing the probability that there exists some SNP, which is the probability that the allele frequency is greater than 0. So I can just say that again because I think it's useful. So imagine I have 10 samples, so I have 20 chromosomes. What you're doing is you're establishing for each of the samples, each of the 10 samples, what is the likelihood of the sample, each of the samples being AA at that site, then AT. And then you're going back through for all of the possible polymorphisms that would be there. So again, you have 10 possible polymorphisms. You would say, what is the probability that this site harbors an AC polymorphism where the C is actually in 3 of those 20 chromosomes? And then you do exactly the same calculation for 4 in the 20 chromosomes. And that probability distribution over all the possible counts of that alternate allele is the allele frequency spectrum estimate, and the probability of the posterior estimate of the probability of being of the frequency be greater than 0 is really the quality score of a SNP call in that site. You're saying, I have very high confidence that there's definitely something segregating here that has an allele frequency greater than 0. That's really everything that's going on with multi-sample calling. And in fact, if you want details, Hang Li, who's here at the Broad and writes a very impressive suite of tools called SAM tools, has also written a very impressive mathematical treatment of this process. So if you really want to dig into the details, I would recommend looking at that. So I thought I would highlight one issue that we've been struggling with for a long time. I think we have pretty reasonable tools to work with. So that quality score I just talked about, the probability that there's something segregating at the site, doesn't differentiate between machine error and real variation. I mean, you're really testing the hypothesis that it's not homozygous reference in all samples at the site. And you can be not homozygous reference for any variety of reasons. I could have a systematic machine artifact there. I could have mismap reads systematically there. Anything that makes you not homozygous reference comes out as in this raw variant list. And then you really have a big problem of separating out true genetic variation in your samples from this potential artifacts. And the potential artifacts are large in size. I mean, we're talking maybe 25% of the raw call sets are artifacts. So this is a figure that illustrates a tool that we've been working on called variant quality score recalibration. What it is a multidimensional Gaussian mixture model that is trained off of a variety of covariates of error. These are 2 key covariates for us: the quality score, that probability estimate of being SNP divided by the amount of reads. So this is really a sort of how much evidence do I have for variation per read that is provided evidence for the variation. And then the strand bias is how much does that variant only appear on one strand of the reads. And what you see is on the left here, these are HapMap sites in reds and dbSNP sites in yellow. And then here are the novel calls that were made for this HiSeq data set. And what you see is that there are 2 distinct clusters. These are heterozygous and homozygous variants. And when you go to the novel sites, there's a lot of new heterozygotes, as you expect. And there's a big pile of stuff that's sort of outside of the cluster. Almost all of the stuff that's outside of these 2 clusters are errors. So I just wanted to quickly point out how to think about Indel calling. So that framework where I talked about where you had likelihoods of the 10 diploid genotypes, and then I have this multi-sample analysis establishing the probability of having exactly I copies of the alternate allele. This applies equally well to not SNPs, but indels or structural variation, whatever you want. As long as you can establish the likelihood for a series of possible genotypes per sample, then you can run that multi-sample analysis. So Indel, which is a tool developed by Kay Alvers and Richard Durbin, is a way of calculating likelihoods, but for indels, not SNPs. And indels are much more complicated to calculate because you really need to realign each read to each possible indel haplotype. Because the nice thing about SNPs is there's 10 fixed genotypes. You can either be AA or you can be TT and anything in between. For indels, you could have a 1 base A, a 2 base A, a 3 base A. It's an unbounded set. So you really have to simultaneously figure out what are the possible alleles that are segregating at a particular site, and then figure out the likelihoods of the reads coming from those putative haplotypes. And then once you do that, you could feed it into the multi-sample SNP / variant caller, which is what we're doing now, and many of the tools are doing. So this stuff is all great, and I think it's critical that we do this. And I just want to give you an idea of what the computational costs are for doing this. I mean, I can stand up in here and say we can do this, but if it took a year to do, this would be very painful. It turns out it takes a long time, but not a year. There's a very clean relationship between the number of samples that you're processing and the amount of CPU time required to process it. So it's a useful equation in case you ever want to do an estimate. There's the x equals y line in the equation. Basically, if you want to process 100 exome samples, these are exomes, it's going to take around 80 CPU days to just do raw local realignment and calling, and then there's some cost for alignment and things like that. So you're talking on the order of 100 days per 100 samples. So this is great. This is why we have a big farm, but it's quite expensive to do these calculations. The other side of that is it's also expensive to store the data. A good estimate here is it takes 2 bytes per base pair generated. So and that's obviously times the targeted area. So here are some numbers about the cost per sample to store exomes, whole genomes, deep or shallow. I think these are actually complete projects down at the bottom. These are more representative of what it's really like to be working with next-gen sequencing data, because it's not just one exome. Hardly anybody works with one exome. The EOMI samples from ESP are 700 exomes. That's a 35 terabyte data set. The 1,000 genomes phase 1 data is whole genome shallow at 60 terabytes. So when you're thinking about next-gen sequencing, you really want to think about, well, I need a lot of CPUs and I need a lot of storage. And this isn't cheap storage. This is high performance, very highly parallelized disk, because you're going to do calculations on that 60 terabytes. It's not archival. It's not like we can put them on DVDs and just write them away. So that's all great. I thought I would move on to, in the small amount of time we have left, talking about a little bit about how to think about quality control. I mean, I'm talking about false positives a little bit. I'm talking about SNP calling. There are many approaches, many SNP callers. Much of this stuff, I think, is people sort of, there's a temptation in this field to sort of just develop whatever statistical algorithm you want to find variation, but not really follow up with differentiating the errors that it finds from the real things that it finds. And so you see many earlier publications of genomes are riddled with what have to be very high false positive rates. And so I think it's very important that everyone think about, if you're developing algorithms or working with data sets, it's very important to understand how do you assess the quality of your output variance. And I think the 4 things that we really think are good metrics, and in fact the last one is, I think, the key metric, is first off, how many calls do you expect to get? You can detect gross under or overcalling simply by saying, I expect 1,000 variants and I saw 15 million, something has gone horribly wrong. If you have genotype chip results, you can use these to assess the genotyping accuracy per sample, as well as the sensitivity of the calling. However, this is a terrible way to assess specificity. You cannot use genotyping chips to really assess specificity, in part because there's an enormous ascertainment on the chips. The chips are only generally run in areas of the genome that are very well behaved. So you're isolating yourself to the areas of the genome where the next-gen sequencing is going to do a very good job. It's really the novel calls that are the dangerous ones. They're in areas that are new, generally not very well understood, and they're full of potential false positives. With respect to that, one key thing is the fraction of calls that are novel. So most calls in the human genome are at sites that dbSNP or the 1,000 genomes is already tagged as a common polymorphism. So if you get a lot of calls that are novel, there's probably something going wrong. And finally, I think the key metric, the one that we really rely on very heavily for determining the accuracy of novel calls is the transition transversion ratio. So the transition transversion ratio is a very simple thing. It's the ratio of AG plus CT polymorphisms over the other 4. And the reason this is interesting is that if I just created random variants, you can see the way this transition matrix is working, I should have a transition transversion ratio of 0.5. I have 2 possible transversions for every transition. So if I just make random SNPs up, I will get a 0.5 transition transversion ratio. However, empirically, due to actual biological effects, the genome-wide transition transversion is 2, and the exome-wide one is 3. So you have an enormous difference between random errors and the actual biological properties of true variation. So deviation from the expectation of 2.1 genome-wide or 3 for an exome is a pretty good indication of how riddled with false positives your data set is. So I think I will just jump over this. Here's an equation for reference more than anything for people that tells you the expected number of polymorphic sites as a function of the number of samples, the targeted area, and the heterozygosity of your samples. This is a simple plot just showing what fraction of the SNPs, that's these 2 lines here, what fraction of SNPs for a European and African individual you would expect to find in the catalog of dbSNP plus 1,000 genomes. And what you see is that most SNPs are known already. So this is a fantastic quality control metric. You should expect for a whole genome around 90 to 95% of your SNPs to be known, and for an exome at least 95%, if not more, already to be in 1,000 genomes and dbSNP. I'm just going to skip over this. I think people are relatively familiar with this. I understand most people are from the GWAS world. I thought I would just put up the key equations / graphics that describe what we are typically referring to as non-reference sensitivity and non-reference discrepancy rates. These are 2 good metrics and are much better than what is called the overall genotype concordance, which you often see in the literature. So just wanted to point out a couple of things about the transition transversion ratio. This is a variety of very high quality call sets from the 1,000 genomes and complete genomics. And it shows the transition transversion ratios for those call sets broken down by known and novel SNPs by the whole genome and the exome. And you can see that the transition transversion ratios are indeed very consistent at the whole genome level and averaging around 3.0 for the exome. So this is useful. But to give you an idea of how much I think we've advanced, there was a caller who I think will remain anonymous right now in the 1,000 Genomes project, in the pilot data, whose novel transition transversion ratio for their official call set for the exome was 0.6. So this is a group that had been working on this for a very long time, and you realize, wow, most of the calls that were coming out were very bad. And we were certainly in very much the same case. You're producing calls that are at 1.5 transition transversion ratio genome-wide, because you don't realize that there are so many false positives. But it's clear from looking at the transition transversion ratio, in retrospect, we should have known better. So just give you an idea of what you should expect to see, what are some numbers, how valuable are these data processing tools. This is a very nice slide put together by Eric Banks. This is showing the value of the Indel realignment and base quality score recalibration on a high-seq data set. So the upper line is the raw reads and then making SNP calls with our current caller. And you can see a variety of things that clearly indicate that this is not a very good call set. The dbSNP rate, as I said, should be 90%. In the raw reads with our caller, you're getting 80%. The transition transversion ratio of the novel genome-wide calls are 1.2. And yet, the HapMap concordance rates and sensitivities are extremely high, 99%. So this is standard problem with SNP calling. Very easy to get very good metrics on chip concordance and still have a lot of bad SNP calls. If you remove the calls that are unique to the raw reads, so those that are not, what we got before we recalibrated and did this multiple sequence realignment, you're really removing tons of junk. 300,000 sites get removed by this process, almost all of which are terrible. But even after doing local realignment and recalibrating all the bases, you still have a lot of obvious false positives in your novel call set. So you still need to be going in and differentiating after even the data processing. You have to have a sort of filtering step, something to remove false positives. This is two ways we've done that. Those are the recalibrated raw calls up there, again, for reference. At 81%, hard filtering. This is some lovingly handcrafted filters that involve strand bias and depth of coverage and allele balance, a variety of things that we've developed for 1,000 genomes. And you see if you apply those filters, you can bring the call set up quite substantially in the transition transversion ratio. It removes mostly novel calls. So it brings your novel call set from 800,000 to 350,000. So it's removing a lot of novel calls. But still, the transition transversion ratio isn't perfect. This new dynamic variant quality score recalibrator that I talked about for one slide is much better at doing this separation, and it brings us up to basically exactly what we would expect. And then you can see validation result. These are de novo mutations. We contributed de novo mutations to the 1,000 Genomes project. You can see the number of false positives. This variant quality score recalibration gives us a much better false positive, true positive trade-off. So very, very important to do these things, to really think hard about removing errors from your data set. One way of thinking about this is in terms of these tranche plots. I like this a lot because it captures simultaneously how good are we at separating true positive variants from false positive variants, and also allows you to understand what type of data sets do you want to work with depending on the needs of your project. So at the top, the very highest quality tranche here is the SNPs that are all, the largest set of novel SNPs that I can find that have a 2.07 transition transversion ratio. So that finds most SNPs. I can then reduce that threshold. I could say, give me all the SNPs that are that sum to a 2.05 transition transversion. And this adds slightly more true positives and slightly more false positives. And I can do this all the way out, sort of through my whole data set, and divide it up in terms of the tranches as accurate as, I guess, as with particularly calibrated trade-offs between true positives and false positives. And what you can see from this is that we do a pretty good job of separating true positives from false positives, but there's still many more true positives in the full data set that we can't separate out effectively from the false positives. So depending on what the goals of your project are, if you're looking for a Mendelian disease, you probably should be working with this 10% false discovery tranche because you don't want to miss anything, and there's not that many false positives to deal with. Versus if you're really looking for a gold standard data set for doing, I don't know, development of new genotype chips, and you want to make sure that absolutely everything you're looking at is real, you would take the highest quality tranche. So just to end, this is actually an answer to the question that was asked earlier. What is the advantage? What is the marginal additional advantage for adding in samples for sequencing? How effective am I at discovering all the sites that I know to be polymorphic in one sample as I add sequencing for just that sample and then more individuals? So the N0 line, which is this red line at the bottom, is what I get if I call an older GA2 data set at 4x coverage, send it through this whole processing pipeline I've described with the variant quality score recalibration, and ask, after that cut to ensure that I have high quality calls, what fraction of the 60x call set that I find? And as you would expect, it's not very good. It's like, I find 20% of the calls. And that's largely because I can't separate true positives from false positives. So I add one more sample. Now it's more likely that I will be able to find a true variant because many of the variants between the red line and the orange line are shared between those two samples. And then I'm also gaining information about which sites are errors and which are not, because I have more data to differentiate true sites from false sites because of these error covariates. So as I add more samples, so I'm going up to 60 samples simultaneously, with just 4x coverage, I'm able to find 75% of the data. And I think the critical thing here is that line there, the first bar, is the fraction of sites where there's at least one alternate allele in the data in the 4x data set we called. So it's an older data set. It doesn't cover the whole spectrum of the genome as the new 100 base pair HiSeq data does. So we can only ever call in that sample, in the NA1287 sample, at that line. So with 60 unrelated individuals, I'm basically maxing out my sensitivity of that data set. I'm just completely limited by sampling at that stage. I just never see the allele. Then the next line up is where we can get to if we impute the genotypes of NA12878 using this all 61 individuals here. So I can use the haplotype structure and the calls that were available at neighboring sites to infer the existence of variants in NA12878 at sites that I don't even have any data at. So I can push that up to 85% or so. And the last line is, I think, the one that I personally think is very exciting, and I certainly will keep my team busy for a long time. The upper line is the fraction of sites where at least there's one alternate allele in any sample at 4x coverage at all sites that are variable in the HiSeq data set. So in principle, we should be able to get to 99% of all the calls with only 60 people in this data set. There exists variation at every site that's truly polymorphic. So it's a surprising thing. With only 60 people, I should be able to get pretty close to complete ascertainment of each individual sample, because most things are shared. So I think I'll skip over this. People can look at that. Basically, I wanted to just end here before we leave. Basically to say that next-gen sequencing is maturing. There's multiple robust technologies. There's several canonical designs I've talked about. There are data processing algorithms and approaches for discovery and genotyping of variants that are all pretty sophisticated now and pretty effective. And I think now you've had a chance to hear about some of the ways we think about quality control of the output. And I couldn't recommend more strongly to have a look at some of the tools in the JTK that do these analyses for you automatically. If you have new data sets, take a look at the quality of the calls that you're working with. Maybe possible to improve them or to add more calls. Maybe you're being too stringent. It's not. You can change the data sets and potentially get yourself more or less, reduce the number of headaches you'll have in the future or add more opportunities. So I just wanted to end with this. We're hiring people. If you have friends who are looking for positions at the Broad, either software engineers or computational biologists, I'd be happy to talk to anyone about this. Thank you.